All right, so speaking of this session is Ashley Nord from Montpellier, and she's going to tell us about single-cell electro-characterization in bacteria.
Great, well, thank you. First of all, thank you to the organizers, because this has been an excellent day so far, and it's my pleasure to be here and tell you a little bit about our small little corner of the biophysics world. So today I'm going to be talking about single-cell electrical characterization in bacteria, which is a new topic to us, albeit using some old techniques. So let me start. For a number of years now, myself and our group, we've been studying the Bacterial Flagellar Motor, which is this molecular machine. It's one of the few examples that you see in nature of a rotary molecular machine, and it's responsible for cellular motility and chemotaxis. So this guy here is E. coli. He's about 2 microns long, and he has an average 5 or 6 of these flagellar filaments coming out at random positions from the cell body. And at the base of each one of those flagella, you find the Bacterial Flagellar Motor, this machine here. And so this machine, it is spanning both of the cell membranes in a gram-negative bacterial cell, as well as the peptidoglycan, that cell wall in the middle. And so this is the inside of the cell here and the outside of the cell here. And so in the inside of the cell, you've got this ring of proteins that is being rotated. And that rotation is coupled up through the inner membrane, connecting it to a structure called the rod. And this rod is basically acting as a central drive train to this motor. It's rotating inside of a ring of proteins that is acting as a bearing, protecting it against the outer membrane and the peptidoglycan. And then that rotation is coupled to the hook, which is this very short but very flexible polymer. And then finally, that is connected to these long flagellar filaments that you see here. And the key players in this machine are the stator units that you see here. So unlike a lot of molecular machines, the bacterial flagellar motor is not driven by ATP. It's driven by the proton motive force, so this electrochemical gradients across the inner membrane. So these stator units, they are attached to the cell wall, to the peptidoglycan, and by attaching there, that opens up ion channels in the middle of them. So they are transporting ions from the periplasm here into the cytoplasm. And because of this electrochemical gradients across the cell membrane, there's a free energy to be had there. And that free energy is transmitted by the stators and converted into mechanical work. So the stators at this interface here are applying a torque to the rotor, and that puts this whole thing into rotation. And the other thing to say here is that, kind of surprisingly, these stators are not a permanent collection of this macromolecular machine. So they can actually exist in 2 different states, either an active state or an inactive state. And you have this continuous and dynamic exchange between those 2 states. And that exchange is actually mechanosensitive. So you see 2 active stators here, but in reality, you have a number of them at the periphery of this motor. The more stators you have around a bacterial flagellar motor, the faster it's spinning. And the larger the viscous load that you have on the flagellum, so if the bacteria starts to swim into a more viscous environment, for example, the more of these stator units you're going to find at the periphery of that motor, augmenting the speed of the motor and helping the bacteria to cope with the conditions. So this is just a slightly more sophisticated version of the same thing, just to tell you that almost all of the parts of this machine we now have high resolution structural information for, which is quite cool. A lot of this has come within the last couple of years. There's been quite a number of surprises that have come out from the resolution of these structures. And in particular, the stators, we think functionally are acting totally different than what the field has thought of for the last 40 years. So it's kind of an exciting time to be studying this machine. But the idea is that even if we have the full structural information, this machine is incredibly dynamic. And so if we want to understand the relationship between structure and function, or anything to do with its dynamics, we have to turn to experiments. So that's what we spend a lot of time doing. And we do that in a number of ways, but one of our favorite techniques is to take living bacterial cells and stick them down to a cover slip. And then we attach a particle to that long flagella that you're not seeing here. And so as the flagella is rotated, it's rotating that particle. So this particle is now acting as a proxy for the rotation of this molecular machine. And so under the microscope, we see something like this. So this has been showed down to hopefully show you that this particle that is attached to a single flagella on a bacterial cell that you see here, it's not just bouncing around, but it's actually going around in a circle. And we can track that particle with nanometer resolution because we're doing this in a holographic microscopy setup. And we know what the interference pattern of this particle with coherent light is. And so we get XY resolution that looks like this. And from this, we can calculate the angular position of the motor. And from that, we can get, obviously, the speed of the motor. So what I'm showing here is kind of an example of the raw data that would come out of one of these experiments where we see the speed as a function of time. We can sit here and watch this machine on the order of hours. And since we know something about the particle that we attach to that flagella, we can also calculate the torque that that motor is producing, or the power that it's producing. So these are kind of the bread and butter experiments that we're doing in order to try and understand the dynamics of this machine. And using pretty simple experiments, we've been able to look at the dynamics on a whole bunch of different levels. So to give you just an idea of the things that we've been looking at the last few years, I told you that these stator units, that there's this dynamic exchange between active and inactive, and that the speed of the motor is proportional to the number of active units we have. So by using that fact, we've used measurements, and in combination with magnetic tweezers, where we can manipulate the motor in different ways, we can actually quantify this dynamic exchange. And what we found is that the lifetime of one of these complexes in the system is proportional to how much force it is applying to the rotor here, and by symmetry, the amount of force that it is pulling on its link to the peptidoglycan. And when we saw this, this was quite curious, right? Because most things in nature, the more force that you apply across a bond, you're going to decrease its lifetime. Whereas here we're seeing the exact opposite thing. So this is something called a catch bond, and this we think is at the heart of the mechanosensitivity of the bacterial flagellar motor, so the ability of bacteria to adapt to its surroundings. More recently, we've done different experiments where we are looking at now at the physics of the hook here. So the hook is this very specific polymer. It's taking a rotation that is happening along this axis, and it's translating it to rotation that is almost orthogonal to it, right? And so on the one hand, it needs to be very flexible in order to do that. But at the same time, the bacterial cell, as it's swimming along, it's subject to a huge amount of hydrodynamic drag. So you want that thing to also be relatively rigid, to not have everything fall apart. So the question was, how do you have a polymer that is at the same time able to fill these two different things? And the answer to that, just by looking at fluctuations of the bead, what we can see is that when the motor is going relatively slowly, when it's not applying very much torque, you see a lot of fluctuations of the bead, and the hook is very flexible. But the moment that the motor starts applying more and more torque, you have a higher and higher bending flexibility of this polymer by about an order of magnitude. So basically, you have a torsionally stiffening polymer. And this is the way that the hook is able to do two things at once. But what I'm going to talk about for the most of today is something entirely different. And that is using measurements of the bacterial flagellar motor in an attempt to try and quantify the dynamics of the proton motive force. So this electrochemical gradients across the inner membrane. This is work that is pretty new to us. This is the first time that we're sharing it. And it's ongoing, and we're still trying to wrap our heads around what we're seeing. So with that, let me introduce and say the proton motive force is kind of the basis for energetics in bacteria, and not only bacteria, in all kingdoms of life. In bacteria, it exists across the inner membrane, right? So anytime you have a lipid bilayer, it's pretty impermeable to protons. So if you end up with more protons on one side than on the other, you have, on the one hand, a chemical potential there due to the delta pH, and on the other hand, an electrical potential due to the gradients of charges. And so the combination of those two things gives you the proton motive force. And this is being actively produced. So in bacteria, it's on the order of about 150 millivolts, at least for E. coli. It's being actively produced by cellular respiration. So you've got respiratory complexes sitting in the inner membrane, and they're pumping protons from this side, from the inside of the cell to the outside. And then at the same time, it's being actively consumed by things like ATP synthase and by the bacterial flagellar motor. So the proton motive force is this source of free energy to drive things that are biologically important, but thermodynamically non-favorable. Okay, so you can think about it as having, a whole bunch of sources, things that are pushing protons from this direction to this direction, and a whole bunch of sinks. And these are existing on copy orders of the hundreds to thousands in the inner membrane. So we know that the proton motive force in bacteria is important for many fundamental processes. It's important for motility, as I've said, and for ATP synthesis. It's crucial for membrane transport and for cell division, and more recently understood as well for antibiotic resistance. And because of all of these things, it was thought for a really long time that the proton motive force was homeostatic, right? So it was something that was homogeneous among a population of bacterial cells, but also looking at a single cell level that it was constant in time. More recently, so this is an image coming out of the lab of Adam Cohen, and they developed this optical sensor that reports on the membrane voltage of bacteria. And when they put this into E. coli, just plain old E. coli, putting it onto a cover slip, you see these transient fluctuations in fluorescence, which are showing transient fluctuations in the membrane voltage. So this was kind of the first time that we saw on the single cell level, this membrane voltage and this proton motive force is not constant, right? And so I would say that this was kind of the birth of the field of bacterial electrophysiology. Since then, we have a lot of reason to know that bacteria are actually using oscillations of their proton motive force as a form of communication. So to give just a single example, this is a biofilm of Bacillus subtilis, and it's actually doing this coordinated oscillation of its proton motive force in order to coordinate the metabolism of that biofilm. So we now know that the proton motive force is very dynamic, even on the single cell level. And from a physicist's perspective, maybe that's something that we should expect, right? You've got hundreds of these sources and hundreds of these sinks, and it's this very non-equilibrium process. And so the question is, how do you go about quantifying it now at the single cell level? You can't turn to typical tools in electrophysiology because patch clamp is simply not going to work for bacteria, right? They're too small. You can't get through the cell membrane. You can't get through the cell wall with that. So you have to come up with different techniques. And probably unsurprisingly, we are going to turn back to the bacterial flagellar motor to do this. And we're not the only ones to think of this or to be pursuing this. But the reason to do so is that it's been known for a long time that the rotational speed of this motor is directly proportional to the proton motive force, right? So that's quite cool because it means that bacteria are have basically fabricated this inbuilt tool to measure this, right? So you've got a local probe sitting in the membrane that reports upon the proton motive force at that specific position. And then ideally, if you want to say something about the dynamics of this, you also need a way to modulate the proton motive force. So for that, we turn to marine bacteria that have this intermembrane protein called Proteorhodopsin, which is a light-driven proton pump. So we take it out of the marine bacteria and we shove it into our E. coli, and it gets expressed, we assume fairly homogeneously in the inner membrane. And so Proteorhodopsin, when you shine it with green light, it starts pumping protons from the cytoplasm into the periplasm. So given that, we expect that when we shine it with green light, we're going to see an increase in speed of that bacterial flagellar motor there. And that's something that we can easily do in our experiments. We just add a green laser to what we're already doing. So when we do that, what we see, so this is the speed of a single bacterial flagellar motor as a function of time. So the gray is showing you the speed. The green is showing you the state of the laser. So up is on and down is off. So you see that the speed of this motor is going at about 50 hertz, 50 revolutions per second. And we turn on the laser and that jumps up to about 90, and we turn off the laser and it goes back to 50 over and over. Now, the bacterial flagellar motor, like most molecular machines, is an intrinsically noisy one, right? So the reason that we want to do this over and over is to build up statistics of these dynamics. So in reality, these traces look something like this. So this is showing you about 50 pulses of the laser. And in reality, we actually do this on the order of about 3,000 pulses of the laser. So now we can take one of these transitions and we can start stacking them one on top of each other to get a good behavior, a good idea of the average behavior. So that's what you're seeing here in black. And what you see is this nice exponential rise to a new steady state and a nice exponential fall back to the previous steady state. And, you know, as a physicist, when you're looking at exponential rises and falls and talking about moving charges around, at least I think about charging a capacitor. So for this reason, we turn to a model of an electrical circuit, and then we're taking inspiration of work that's been done previously. And so in this model, we've got the cell membrane, which is acting as a capacitor, right? It's fairly impermeable to these ions. We've got cellular respiration, which is acting as a battery for the cell, albeit not a permanent, not a perfect battery. It's got an internal resistance. And then you've got proteorhodopsin, which is acting as a second battery for the cell. But proteorhodopsin is only operating when we shine the laser on it. So we'll put a switch in the circuit here, such that when the laser is on, it's like closing that switch. When the laser is off, it's like opening it back up. you've got the bacterial flagellar motor up here. So you've got ions slowing this direction, crossing the membrane through the bacterial flagellar motor. It's basically acting as an electromechanical voltmeter for the system, right? So we're measuring the speed and we're using that as a proxy for the voltage. And then you've got all sorts of other sinks in the system like ATP synthase that are also consuming it. Okay, so using this mathematical framework, we can predict what should happen when we turn on the laser or we close this switch. We expect an exponential rise, and when we turn off the laser or open that switch, an exponential fallback. And this circuit isn't too difficult or isn't too complicated, so we can actually have an analytical solution for what happens there. It's a little messy, so I'm not going to put it up, but what I will show you is that we can calculate the time constants that we expect for this behavior. And I write them down here just to show you that there's one very specific prediction that comes out of this system, and that is the time that it takes to discharge the capacitor, discharge the system when you open the switch should be larger than the time that it takes to charge this system back up. And the reason for that is because you're opening the switch and taking a resistor out of the system. Okay, so if we take that mathematical framework and go back to our data, we can fit the individual transitions, right, with these single exponentials. We can pull out the parameters from that, and what we see immediately is indeed this the time to discharge the system is larger than to charge the system. So that's already quite cool. And we can fit that model to our average behavior of the system, and we find kind of an impressively good fit. So a simple circuit model seems to reproduce this system pretty well. And then we can take those parameters and we can go back, and ultimately our goal is to actually say something about the electrical characteristics of the system. In particular, we can say something about the currents that is flowing through the bacterial flagellar motor, which is cool because this is the only biophysical technique that has anything to say about this at this point. Okay, so the next thing that we wanted to play around with was we wondered what would happen if we produced the proton motive force in spot A, what happens a very long distance away. So to do that, we exaggerated and created very long bacterial cells, so about 20 micron long, so about 10 times the usual size of a bacterial cell. And now we take our laser and we just shine one end of the cell, and we measure the speed of the motor on the other side. And so the question is, when you turn the laser on here, what happens over here? And generally what we see, so now I'm showing you directly the average of about 3,500 of these transitions. But when we turn the laser on, you immediately see the single exponential rise, exactly as you saw before, and the single exponential fall. So in other words, this motor that's sitting here, it's as if it was right underneath this laser. And we can do kind of the same thing, but in a different way, but now go and takes a while to find, but to search and find a bacterial cell with a motor on each end of the cell and shine only one of those motors, right? And the reactions that you see, so blue is the motor right under the laser and red is the one that's far away. You can see that qualitatively, the response is more or less the same. These two motors you see right away, they've got inherently different speeds, which is not surprising because we see a lot of heterogeneity even on a single bacterial cell. But what we see is that if you normalize these two curves to their steady state speed, you end up with that, which the first time that we saw that, it was quite impressive, right? Because we're saying that the proton motive force, not only does it travel far, so far meaning many lengths of a single bacterial cell, it travels fast, meaning that the moment that we turn the laser on, we see a response, not only from this motor, but from this motor. But on top of that, it travels with very little loss, right? So it's like as if all of the proton motive force that this motor sees is also seen at the other end of the cell. So with that, let me conclude and say that it's not maybe the most intuitive tool, but we think that the bacterial flagellar motor is this fast and very local probe of the proton motive force in bacteria, and it allows us to do very high resolution electrophysiology of these cells. We've got a simple mathematical circuit that describes pretty well our experiments. We see that the proton motive force, it travels far, it travels fast, and with little loss. And one of the conclusions of this is that the proton motive force on the scale of a single bacterial cell, it's actually homogeneous, which was not obvious to us a priori, right? Because we were thinking of this thing as a whole bunch of sources and a whole bunch of sinks scattered randomly in the inner membrane. it's not obvious that you would have a homogeneous proton motive force. And in fact, people are thinking that, for example, the respiratory complexes, we see them spatially localized during different types of metabolism in the cell. So it was thought that they were doing that in order to produce more proton motive force on one end or the other. What we're saying is that no matter where it's produced, it's homogeneous on the scale of the bacterial cell. And I didn't go into it, but our explanation, or the way that we understand this, is that it's not actually to do with proton diffusion, right? Naively, when we started out, we were thinking that if you pump a proton across this membrane, you have to wait for that proton to go from A to B before you see a response at B. But proton diffusion, it's really fast, but is actually way too slow to explain what we see here. And so what we think we're seeing is actually the other component of the proton motive force, the propagation of the membrane potential, that is responsible for the response in the cells. So with that, let me thank a whole bunch of people that contributed to this work, in particular Anais, who is a very talented PhD student who did all of the experiments that you saw there, as well as her co-supervisor, Francesco, and of course, all of the people that pay for us to keep the lasers on and the bacteria spinning.
Okay. So you talked about the other battery in the cell being the respiration. So could we use these experiments to also quantify the energy of respiration? And second question related to that is, if the substrate for respiration is changed, like the glucose is changed, do you think that the rotation voltage varies?
Yeah, two good questions. So our ultimate goal is to be able to pull out all of the parameters of that, including, yeah, indeed, that of respiration. It's going to be a little bit difficult because obviously there's a lot of parameters in this model. And so we need to be able to vary things in a systematic way in order to do that well. But yes, and with respect to what will happen with different forms of metabolism, to my knowledge, no one's tried, but yeah, I would expect it to be different. For example, one of the things that I didn't mention, but in our experiments, we need to actually deprive the cells of oxygen a little bit in order to see this response. And the reason for that is that if this battery is too strong, basically it's outpowering this one.
So very cool talk, and not my area, but and maybe a dumb question, but if it's not if protein diffusion is too slow and voltage is propagating it, will this basically increase the concentration of all other positively charged species on the other side because they're getting pushed by the voltage, or like, will weird things happen?
Yeah, no, it's a great question. So to be honest, we haven't totally wrapped our head around all of these ideas yet, so take it with a grain of salt. But what I can tell you is that we've done the experiment of two bacterial cells side by side, right? And if you illuminate one bacterial cell, you see a response of all of the motors on that bacterial cell, but not to the bacterial cell next to it. So that tells us that the proton motive force that is propagating is not propagating to neighboring cells. And that is also true even if the cells are touching. That said, it sort of disagrees. You know, there's already really nice work done with bacteria showing that they can open, for example, potassium channels or calcium channels. And that we know that those positive ions do propagate for long distances. So how we explain the differences, we're not quite sure yet. It may be a case that protons are staying on the membrane of the bacterial cell.
Thank you.
Exciting results. What's the wavelength of the laser, and what's the absorption coefficient of the medium that you're using in which the bacteria is?
Let's see, the wavelength of the laser is green, I believe 552.
532.
And the absorption of the media. So this is just in a bacterial medium motility buffer, so similar to water.
So why I'm asking this question is because it seems your rise time is of the order of 50 to 100 microsecond, and fall time is also of that order. That seems to be more like density or phase change in a material when it absorbs heat from a laser. So what I'm trying to say is that if you have a fan, for example, if you have a fan which is running in air with certain voltage being put in, if you put that fan in water, it will start running slower. So in your case, if you have a bacterial flagella which is moving, say your medium is water, and then if I suddenly heat up the medium and I change the density and make it lighter, then the same flagella with the same amount of voltage would be running faster. It's like getting the fan out of the water and putting it in air. So how does that differ?
Interesting. I'm gonna have to think about this. So first of all, our characteristics times are on the order of like 10 to tens of milliseconds, so not quite as fast as what you say. One answer to that might be, we've done the controls. If you don't put proteorhodopsin in the bacterial cell and you just shine these cells with laser light, you don't see a change in speed. Aside from that, you might be onto something that I'm not quite comprehending, but we should talk.
Sure. Thank you. Amazing talk, amazing data. Surely protons themselves don't have to move that distance. There's just a chain of waters, and it doesn't take much of ordering to have a very fast chain where one on at one point and it's one off at the other, 10 microns away.
Yeah, so indeed, I mean, you've got the Grotius mechanism such that you only need to break hydrogen bonds in order to move charges, so to speak. Even that, as far as we understand, is not fast enough unless there's something very interesting going on in the area between the two membranes here. You might have extreme ordering. I'd be interested to understand how many orders of magnitude that would change the speed.